Welcome to this lecture on animal behavior. Today, we are going to explore a truly fascinating topic known as stimulus filtering. Imagine the sheer amount of sensory information an animal is exposed to in the wild. Every rustling leaf, every shadow, every scent. How does an animal know what to pay attention to and what to ignore? Understanding this mechanism is absolutely crucial because it is the very foundation of how animals survive, hunt, and avoid predators in a highly chaotic environment. Let us dive in and uncover how their nervous systems perform this incredible feat. To understand why this topic matters, let us start with a familiar scenario. Imagine you are standing in a crowded, noisy railway station. Despite the overwhelming wall of sound, if someone calls your name, you instantly turn your head. Your brain has automatically tuned out the useless noise and focused entirely on that one important piece of information. In the wild, animals face this exact challenge every single second. They are constantly bombarded with sights, sounds, smells, and vibrations. If they tried to consciously process every single detail, their brains would completely overload. They need a way to cut through the noise. Now that we understand the problem of sensory overload, let us define exactly what stimulus filtering is. In animal behavior, stimulus filtering is the remarkable ability of an animal's nervous system to selectively ignore vast amounts of sensory information while extracting only the biologically relevant cues. It is a biological shortcut. By filtering out the useless data, the animal ensures that its limited cognitive resources are dedicated entirely to events that actually matter for survival, such as finding a mate, locating food, or escaping a predator. It is not just about hearing or seeing, it is about prioritizing what is important. Building on that definition, let us look at how this filtering actually happens inside the body. This process can occur at two different levels. The first is peripheral filtering, which happens right at the sensory organs. For example, an ear might be physically built to only detect a specific frequency of sound, completely ignoring all other frequencies before the brain even gets involved. The second level is central filtering, which happens deeper within the brain. Here, the brain might receive a wide range of signals, but it uses specialized neural circuits to actively suppress the irrelevant ones. Both mechanisms work together seamlessly. Understanding these mechanisms leads us to an important question. Why is this so absolutely critical for survival? The answer comes down to energy and time. Processing sensory information requires a massive amount of metabolic energy. If a frog had to consciously analyze every leaf blowing in the wind, it would quickly exhaust its energy reserves. More importantly, processing all that useless data takes time. In the wild, a delay of just a few milliseconds can mean the difference between life and death. Stimulus filtering allows animals to react with lightning speed, keeping them alive in unforgiving environments. Now that we have explored the biological concepts, let us look at how scientists measure this mathematically. Biologists use signal detection theory to quantify an animal's filtering ability. The core metric is the sensitivity index, called D prime. The formula is D prime equals mu s minus mu n, all divided by sigma. Let us break that down. Mu s is the average intensity of the actual signal, like a predator's sound. Mu n is the background noise, like wind. Sigma is the environmental variation, or how unpredictable the noise is. If the signal, mu s, increases, D prime goes up, meaning easier detection. If background noise, mu n, increases, d' goes down, making filtering much harder. Before we move on, let us see this formula in action with a concrete example. Consider a noctuid moth trying to survive the night by listening for the high-frequency squeaks of a hunting bat. We are given that the bat's signal creates a neural response of 45 millivolts. The background forest noise is 15 millivolts, and the environmental variation is 5 millivolts. We want to find the sensitivity index. We substitute these values into our formula. 45 minus 15 gives us a difference of 30. Dividing 30 by 5 gives us a final d' value of 6. Physically, a value of 6 is extremely high, meaning the moth's auditory system perfectly filters out the forest noise to instantly detect the bat. Having calculated the sensitivity index, let us visualize exactly what that means. On this graph, the horizontal axis represents the intensity of the neural response, and the vertical axis represents probability. You see two bell curves. The blue curve on the left represents the background noise, the irrelevant stimuli. The red curve on the right represents the actual biological signal mixed with that noise. The distance between the peaks of these two curves is our d' value. The larger the d', the further apart these curves are, meaning the animal can easily tell the difference between a harmless gust of wind and a deadly predator, crossing the neural threshold. Now that we have seen an auditory example, let us shift our focus to a fascinating visual example. Toads have a highly specialized visual system that acts as a strict stimulus filter. When hunting for food, they do not just strike at anything. They are hardwired to only attack small, elongated objects that are moving horizontally, like a worm. If an object is too large, or if it is completely stationary, the toad's brain completely ignores it. Even though the toad's eyes physically see the object, the brain filters it out as irrelevant background information, saving the toad from wasting energy on non-food items. To understand how the toad makes this split-second decision, we can model its neural activation mathematically. The formula is simple. A equals S times V. Here, A represents the total neural activation inside the toad's visual cortex. S is the size factor of the object being viewed, and V is the velocity or speed of that object. The toad has an internal biological threshold, which we will call T, set at 50 units. If the size and speed of an object combine to create an activation A that is greater than 50, the toad strikes. If either the size or speed drops, lowering A below 50, the stimulus is filtered out. Let us apply this formula to a real scenario. Imagine a leaf falling in front of the toad. We are given that the leaf has a size factor of 15 and a slow velocity of 2. We need to determine if the toad will attack. We multiply the size, 15, by the velocity, 2, which gives us a neural activation of 30 units. We then compare this to the toad's critical threshold of 50. Because 30 is less than 50, the visual system filters this information out entirely. The physical meaning here is that the toad's brain categorizes the leaf as irrelevant background noise, and absolutely no action is taken. Now, let us contrast the leaf with a crawling worm. We are given that the worm has a smaller size factor of 8, but a much higher velocity of 10. Again, we calculate the neural activation by multiplying 8 by 10, which results in 80 units. We compare this to our threshold of 50. Since 80 is significantly greater than 50, this stimulus successfully passes through the toad's neural filter. Physically, this means the toad's brain recognizes this specific combination of size and movement as highly relevant prey, prompting the toad to immediately shoot out its tongue and catch the worm. This brings us to the end of our exploration. So let us summarize what we have learned. First, stimulus filtering is a vital evolutionary adaptation that prevents animals from being overwhelmed by chaotic sensory data. Second, by using specialized organs and neural circuits, animals effectively mute irrelevant background noise and amplify the signals that truly matter. Finally, whether it is a moth listening for a bat or a toad watching for a worm, this biological filtering ensures that energy is conserved and reaction times are minimized. You have now mastered the core concepts and mathematics behind this incredible biological system.